Excellent. All right. There we go. Hope everyone can see those. First of all, very much thank you to Amy for inviting me to be here today, and thank you all of you for starting your day with me. I hope you know, by the end of the talk, you'll have a sort of understanding of the work we're doing. I am going to talk about lupus, but I think this is a good sort of a case that's sort of a representative of the kind of work that I'm trying to do, which is sort of surveillance work for complex disease and medical record systems. And quickly, this is kind of a play in 4 acts. I'll talk a little bit about some basic introductions, tell you about a couple of studies I've been working on that sort of walk us through the work we've been doing with lupus, and sort of a little bit about what is next. And one thing I will say is I totally am open to questions during the time I'm talking if folks want to, but you might have to speak up because I'm not so good at monitoring the chat while I'm doing these things. So just quickly, who am I? Amy told you a little bit. I am a bench-trained immunologist, and I did my bench training sort of towards the end of the 90s, and so got really excited about genomics. The human genome was coming, and what was clear, even though I was a bench scientist, was that this was really important stuff, but we weren't really going to know how to manage the data. And I got really excited about that and decided to get my master's degree in computer science so that I could help figure out how to do that. Along the way, I got a little bit distracted and ended up in a startup company focused on microbial genomics. So I got my genomics, but I got it in a little bit different aegis. But this was actually also where I started to put my master's in science into sort of practice for informatics and figure out, well, how do we build systems to sort of translate complex information into something that's easily used by bench biologists and translational scientists in that space. And after I'd been there for a while, I had the opportunity to come back to Northwestern and work with Abel Coe, who was setting up the Chicago Health IT Regional Extension Center at the dawn of meaningful use, to help sort of get electronic health records distributed across the country. And it was very shortly after that that it occurred to me, maybe I can go back to my roots and figure out now how can we use this data to understand human immune disease. And like I say, I was a mouse immunologist, and mice are great. But one of the things about mice is they're not people. And unfortunately, while we can do some low-level mechanistic things to understand what's going on with people, what becomes really difficult is many of these kinds of chronic diseases, such as autoimmune disease, even some kinds of cancer. It's really hard to reproduce these in mice in ways that allow us to really understand all the nuances of the human condition. And so there is a motion, particularly in immunology right now, to figure out like how we do a little bit more to understand human immunity. And I love this slide because it's actually a slide that comes out of a recent review article by a relatively well-known immunologist, where he's trying to sort of advocate for human immunology. And you kind of notice that there's all these different kinds of technologies that we can use to sort of understand the human immune system. And at the very bottom, there's a note of clinical metadata. And this always makes me feel good that at some level clinical data is at the bottom. But the fact that it's labeled metadata and the fact that it isn't kind of, I think, interwoven a little bit more, it gives me pause and makes me hope that this is a place where I can actually sort of help people see that there's so much powerful information in our clinical data sets, particularly with what's going on with what physicians are capturing and care teams are capturing, but also the labs, which I think are really sort of an important and untapped resource. And I throw this out here. I hope that by starting with the immune system, I'm reminding you how cool they are, and that this will sort of help you get into the talk a little bit better. And that's where I'm going to move to what part of the immune system am I looking at. And my connection to lupus is a little bit serendipitous, like a lot of good things are. I met a researcher here. She's a clinician and lupus expert who focus, and her focus is really on sort of improving care from a population health level. And as it turns out, lupus is actually a really good model system too for trying to, from an informatics perspective, because of its complexity. So in terms of developing phenotyping and also developing strategies for population health. So what is it? It's a very complex multi-system autoimmune disease. It has highly variable presentation. And the chart on the left kind of gives you all the different things that can present lupus, as well as a sort of percent like rate that it occurs in people. You can see that there are some things that occur constantly, like this positive anti-nuclear antibody test. And there are other things, the vast majority of things, right, that maybe only occur in 20 or 30% of people. And as a result of this, it can be a really difficult disease to identify. But it's an important disease. Even though it's rare, it has a higher prevalence in women and women of color, who often experience more health disparities anyways. And what's true also about lupus is that it has long-term complications that can be very severe and life-threatening. In particular, it can have severe kidney damage and stage renal disease. And one of the things that's true about this too is that early identification could lead to earlier intervention, and that reduces the disease damage these patients experience. And there's a lot of studies that show that existing disease damage actually is a predictor of future disease damage. So again, the earlier we find folks, better. Now, the first thing I'm going to tell you is a study that we worked on about 5 years ago. And it was focused on, as things are, sometimes you get projects that are based on serendipity and access to reagents. And in this case, I had access to a really unique data set that was sort of an integrated medical record data set from across Chicago. And so I decided that one of the things that would be really interesting to understand is whether care fragmentation, which meant having care at more than one health delivery site, whether it could impact the outcomes for people with lupus. And so our goals were pretty simple. First, we needed to identify patients in the data set. And then assess the rate of care fragmentation and sort of try to look at the relationship between fragmentation and outcomes. And just the data set we were working with, this is the Chicago HealthLink data repository. It is unfortunately no longer easily available, but it was work of a colleague of mine, Abel Coe, where he was focusing on sort of privacy preserving record linkage. And what this is a de-identified, deduplicated, merged record set of about 2.3 million people from 7 organizations across the city of Chicago. The data in there is from about 2006 to about 2012, and its ages of primarily adults. What's neat about this data set is they validated it, and it is actually pretty population representative with regards to the 2010 census, with the obvious caveats of medical record data that younger people tend to be less well represented, and so forth. And obviously you're only tracking people who are actually getting clinical care. So the first thing we did was essentially take a look at the overall population and sort of sorted out who had fragmented care and who didn't. And we found that about 20% of our population, I probably can't see that, but like had fragmented care. Notably, when we looked at sort of racial demographics, and these are, to be clear, these are the OMB categories. These are not patient self-report. What we found was sort of a skewing in the fragmented care of patients who had Black or African ancestry. We also found that there was a skewing towards public insurance. So the reason I'm showing you this is I want to make it clear that fragmentation really isn't the only important variable that we're looking at here. There are race, and we know that race and ethnicity play into fragmentation issues. We also know that insurance has an impact on how people receive care. And this was sort of the start of the observations that got us really thinking about why care fragmentation would be important. So essentially what we did here is we identified a collection of comorbidities that are associated with lupus, including serious infections, end-stage renal disease, kidney disease, cardiovascular disease, and stroke. And the first thing we did is we identified the patients with lupus, and we did this very trivial definition to some extent. It was based on, at the time, sort of what was being used for claims data, which was 3 or more diagnoses of lupus 3 months apart. Then we used, again, primarily diagnosis codes to identify the different infections and the different conditions we were looking at. And what you see here is no fragmentation, patients with no fragmentation in the purple bars, patients with fragmentation are in the blue bars. And hopefully the trend, I think, should be more important than the actual specifics here. And you can see that pretty much in every case, patients with fragmented care had a higher rate of the negative outcome. And this was statistically significant. And so we wanted to do a little bit of modeling to understand what was going on here. Was this actually something that was really resultant from fragmentation, or was it resultant from one of those other variables, race, insurance, some other access to care issue that might be, might have an impact on this? And this slide, a little bit complex. Focus with me on the sort of the dark purple bars on the right-hand side. And what we're looking at here in this case is we did some multivariate modeling to understand how care fragmentation had a relationship with outcomes. And they're pretty much, the trend was always the same. Our reference population here is white patients with private insurance and no fragmentation. And as you can see, this is always the first. If we compare African-American patients with private insurance, they still have higher odds of a worse outcome. But when we add in, for instance, fragmented care to the private insurance mix, now the odds ratio gets worse. And this is more or less true for also white patients on public insurance. When fragmentation is present, the odds ratio of them having the worst outcome gets higher. And finally, when we look at African-American patients on public insurance, and then we add in fragmentation into the mix again, you can see that the odds ratio gets much higher. And so our conclusions for this were essentially that while fragmentation was not the only driver, certainly, of these negative outcomes, it was certainly associated, and it seemed to have an additive effect with race and a sort of insurance access. And it's honestly been this sort of finding that's been driving a lot of my work in lupus. Like, how do we sort it out? And so the next two things I'm going to show you kind of get at two pieces of what we're trying to look at. I mean, the first piece is, well, maybe there's just something going on with this particular patient population. Maybe it's really still all patient variables that are driving this, and it's particular types of disease manifestation that's occurring that's increasing the rate of fragmented care. And so it looks like something, even though it's like fragmentation, even though it's the patient. The other situation is maybe it's actually something related to what's going on with the data. If, for instance, maybe there's a patients with fragmented care, the clinical teams can only see a certain part of the data, and maybe they're only able to make the quality of decision making is lower because they don't have all the information. So what you'll see in the next couple of slides, the next two stories are my attempts to sort of both get at characterizing lupus and then also taking a look at the impact of care at multiple sites. So the first thing is was a long and sort of exhausting project, but was actually super amazing and fun. And we had to figure out, like, well, okay, we know we can identify lupus. There's actually been several phenotypes developed to identify lupus, the most famous of which is April Barnato's from Vanderbilt. But what we were kind of going back to that notion, first of all, that sometimes it's hard to identify people with lupus, and then trying to understand what attributes of lupus might be associated with more severe outcomes. We thought it'd be really important to see if we could figure out whether we could identify attributes of lupus that would help us identify subpopulations, as well as potentially do some early identification. And so our goal was to essentially find a way to create a computational phenotype for lupus symptoms that were based on clinical classification criteria, which I'll discuss a little bit more, and then figure out whether we could actually use these criteria and find them in clinical data at a single site, which was Northwestern medicine. Just a quick sort of overview for those of you who might not be familiar with them. There are actually 3 classification criteria for lupus, and classification criteria really broadly are essentially a set of criteria used to describe patients for research studies. They're really awesome because generally the development of these are, through extensive Delphi processes with experts. So they're not just one person trying to sort of figure out the universe. They're actually like a community of rheumatologists who have spent a lot of time thinking about how we define lupus and what makes definite lupus. Long story short, I'm only going to show you the results from one classification criteria system, but we did look at all three of them. But to make things a little easier, and this is the one we focused on. I'll just talk about this one. So this one is a Systemic Lupus International Collaborating Clinics Clinical Classification Criteria System. SLIC is as it's called, is a system that basically has 17 attributes that it defines, and they're in two domains. There's a clinical domain and an immunological domain, and most of them are in the clinical domain. The immunologic ones are your labs, not surprisingly, and the clinical ones are generally defined by diagnosis codes, procedure codes, and so forth. To satisfy the classification criteria, you need to identify one in each domain, and ideally, you need 4 or more. And if you have 4 or more, we would classify a patient as having definite lupus, in addition to knowing what attributes it satisfies. You can see this is relatively non-specific in some ways, in the sense that it's not that any particular 3 or 4 immunological criteria or clinical criteria have to be identified. So it allows us to sort of get at the characterization of the patients without, sort of saying it's only these 3 things that it can be, or what have you. It gives us a broader picture of what's going on with patients, and as you can see, might create a natural sort of machine learning environment to work with. So how do we do this? Well, the first thing we did was essentially built off resources that had already been developed here. We have a registry here called the Chicago Lupus Database. It's kind of a physician get validated gold standard. All these patients have been adjudicated for the classification criteria. There's almost 900 of them, or there were at the time we did this work. We then determined how many of these patients had data in our enterprise data warehouse and had 3 or more visits. And we just did that to make sure they had reasonable data depth. We found that we had about 500 patients in that category. And to make sure that we had a control set, right, we had a group of patients who had lupus and had been adjudicated for these criteria. We also need to make sure we had a group of patients that didn't have lupus and had also been adjudicated for the criteria. And so there was a little bit of a heroic effort on the part of a number of medical students working on the team. We identified a group of patients in general medicine who were over 18, had 4 or more visits, and no lupus diagnosis. We called these healthy controls, but the truth of the matter is they mostly just didn't have to have lupus. They did not necessarily have to be without other diseases. Our algorithm design process was pretty straightforward. We looked for diagnosis codes, lab results, and procedures that were associated with the different types of criteria that we were defining. And to be clear, this was only something we did in structured data. The reason being is that we were concerned about portability of the algorithms across organizations, and we were concerned that doing things like NLP would reduce that. And we wanted to see how far we could get with just structured data alone. And I'll tell a little bit more about that. And then finally, we just obviously assessed these algorithms over our population, both cases and controls, and we refined as we went. And improve those to the point where we could not make them any better. And this slide here is something of an eye chart, but it's also my attempt to sort of describe the work that we did in attribute detection. So, and the sensitivity and the specificity of the individual attributes in here. What you'll see here is in this slide, in the graph, the clinical attributes are in red. The blue attributes are the immunologic attributes, which are lab defined. And so the bars represent the sensitivity, the dots represent the specificity. I think what hopefully is clear without looking too much at the labels is that actually we had a pretty wide range of sensitivities. So our ability to detect certain attributes. Specificities were a little bit more consistent, perhaps not surprising because, when things are there, it's easy to not find them not being there. But one thing I hope kind of draws your eye is actually the things that have tended to have the highest sensitivity actually tended to be our laboratory attributes, whereas some of the things such as I think oral ulcers, photosensitivity, these tended to have a little bit lower, a lot lower sensitivity, and a large part that has to do with the fact that some of these things are not diagnosed for. So this was a really important learning process in this for us, was that there's a lot of these clinical attributes that are actually really important that are really hard to find documented in medical record data without doing text processing. Also, we noticed that some of the lab results, that things were lab-based, like leukopenia, that you see sort of on the far left. They have high sensitivity, but they actually have pretty marginal specificity. And so important, but perhaps we need to refine for the future if we really want to make it work for lupus. One last bit is that if you look for the anti-nuclear antibody, which is kind of the bellwether test for lupus, what you notice is we've only got a sensitivity of about 70%. And this is an important thing to call out because you can't find what's documented in the notes again. And a lot of patients will only receive this test once or twice in their history of disease. It's not something that rheumatologists run regularly over and over again. So again, probably, even though this is a super important test, there's probably a lot of historic information and notes that we didn't gather. But that said, how did this work to sort of characterize people with lupus? Actually, not so bad, surprisingly enough, even with all the problems with sensitivity. When we sort of assess the criteria as a whole, the criteria set as a whole over the population, we found that about 76% of our cases we could identify, and we didn't identify hardly any of our controls. So it's only about 0.6% of our controls did we sort of satisfy the criteria. And when we reviewed those, most of them turned out to not be lupus. And again, this had to do with some of the like leukopenia attributes and some of the things that were sensitive but not particularly specific. So we were actually overall pretty pleased with that as a foundation for next steps. And just a sidebar, that when we looked at the other two classification criteria, neither of them performed quite as well as this one. And we think that has to do with, in large part, how reliant this particular criteria is on labs as opposed to sort of clinically described criteria. So that was fun. We got a set of tools that we could use, albeit with some caveats. But one of the criticisms that we got when we published this from the reviewers was, well, this is really great, but you did this in this really closed environment. And if you're really focused on thinking about how you can find people, in the population, or if you're going to use this to help identify potential misdiagnoses, this sort of highly controlled case control set is not really helping you. So we wanted to do, the next thing is we wanted to figure out what would happen if we looked at, we tried to use a more general patient population in which to identify try to identify people, characterize people. And so the next and the last sort of full act of this story is just sort of looking at this criteria set and how it performed in a multi-site data set. And this is sort of where I'm really excited to be going right now, which is really thinking about surveillance, both for lupus and some other work that I'm doing. And so what we wanted to do is, like, we'd shown that in general, these classification criteria could define a cohort of patients that we knew had lupus, and perhaps not a surprising result. But we really wanted to see, like, okay, if we assess this classification criteria over a much broader general cohort and one that includes data from places outside of Northwestern medicine, did satisfying the classification criteria actually, was that associated with having lupus? Or did these classification criteria just pick up lots of stuff when we get out into the real world? And so that's kind of, that's the first part of this. And the second piece of this is because we were in a multi-site data set, was we thought, oh, maybe now we can see, what happens, how much more data accrues when you have an additional site. So if your care is fragmented, how much is at your primary site? How much is at your secondary site? And could that potentially make a difference for clinical care? So the next, again, a little bit more serendipity. I'm again fortunate, like I said, I share a wall with Abel Coe, and he has been the principal investigator for the Chicago Area Patient-Centered Outcomes Research Network. And CAPRICORN, which was formerly part of the CORNET, is really an incredible resource. It covers 12 different health systems and even more sort of local sort of hospital organizations. And it has about 12 million records from patients from 2012 to present. What's really also cool about this, I just want to say, is that these clinical systems really have overlapping catchments. And so if you really want to study care fragmentation, your goal is to sort of look for, like, to identify patients and have as many of their health delivery organizations as you can in the data set. And while CAPRICORN isn't perfect, it doesn't include everybody, and it certainly misses small practices. It really is probably one of the best tools around for exploring sort of this issue of finding data for patients in multiple places. And it's also very diverse. In other words, this group does include academic medical centers such as Northwestern, but Cook County Health and Hospital Systems is our public hospital, takes all comers. We have the Alliance Chicago, which is a community health center network. And there's, for instance, like North Shore, which is a little bit more community hospital organization and Veterans Administration. So pretty broad group, pretty broad patient population. So we adapted the tools to the CORNET Common Data Model, which everybody is running under the hood. And this is, again, the other reason why I wanted to stick with structured data, because there are no, the CORNET at this point didn't support any sort of text data storage. And what we did was our inclusion, the way we set this study up was we said that every adult over 18 years of age was included. And they had to, we select our population by requiring that they had one or more of our classification criteria at least one site in CAPRICORN. And again, all the data is from 2012 to 2020. What do we collect? All the primary demographics, insurance status, the number of care delivery sites, the number of times patient was diagnosed with lupus or lupus nephritis, and the presence or absence of these clinical or laboratory attributes. Now, one thing you should know is that investigators aren't returned information about which specific sites. Like, I know how many different sites, I had a line level data set, but I don't know which site provided which data, and that's sort of how the network works. So I ended up with a final data set that was deduplicated and de-identified, and was a line level data set for 7 sites, and actually included 1.2 million patients. So felt like a nice, robust data set. And I want to just point out, all this next work you're going to see was done by a really rockstar student in my lab, Noah Forrest. He's an MD, Ph.D. This is our characterization of the CAPRICORN data set. So again, we had a total of 1.2 million patients, roughly. Of those, about 6,500 had lupus or had one or more lupus diagnosis codes. It actually had 3 or more lupus diagnosis codes, I think, how we did this. Not surprisingly, if you look at the sex ratios, lupus skews heavily female, as it should. Age, not too different across the populations. You'll see that there's a heavier skew for Black or African-American race designation in our lupus population, again, as we would expect. And we also see that there's a slightly higher public payer. Again, not entirely unusual, because lupus patients often have some disability issues. And when we get down to sort of visits, what we see is that lupus patients definitely higher users of the healthcare system. They have averaged more outpatient, inpatient, and ED. Although you'll notice that this, sort of the error on this is pretty high, and probably the range on this is very broad, and that's consistent with the fact that the disease is different scopes for people. And interestingly enough, we saw for lupus, I don't think this is statistically significant, slightly few more sites visited. But on average, for those patients with no lupus, about 1 site. For lupus, 1.3 sites. So that was the basic demographics. So it's just, again, pretty much the side of stuff we saw when we looked in HealthLink. Now we took a look at sort of, okay, what happens when we find, look at all these attributes in the lupus cohort. And so again, we separated these by people who didn't have a diagnosis of lupus and people who did, and sort of looked at the sort of the rate of presentation of each of the attributes in both populations. And you can see some things that, like, for instance, I told you about that leukopenia one that shows up pretty strongly in both cohorts. But you can see in general, the lupus labs are less prominent in our no lupus population. But if you looked at overall satisfaction of the classification criteria, we saw a very small percentage, about 0.6%, who satisfied the criteria. This is actually not so different from what we saw when we did our highly selected controls. Whereas about 40% of the lupus patients satisfied the classification criteria and were identified as definite lupus. You might say, wow, this is actually pretty underwhelming because you have all these patients who have multiple diagnoses of lupus, yet they don't satisfy the classification criteria. And you wouldn't entirely be wrong about that. But what I'm excited about at this point is that what we are seeing is that this classification criteria set is not sort of just, there's not a high rate of these attributes showing up in our general population. So it gives us some hope that we actually could continue to use it for perhaps early identification. We can use it in more general data sets to sort of figure out if we can, identify probable lupus for prioritizing care. And again, there's lots of room for refinement of these classification criteria, particularly the ones that are not diagnosed for regularly in clinical care, like, again, oral ulcers or photosensitivity, or arthritis is another one which is important. But often because the lupus diagnosis is there, doctors won't use those other codes except in notes. But this slide, so this is actually, I'll be honest with you, it's my favorite slide in the whole talk. It's because it proves something that's obvious, but also important. And what we wanted to do essentially is look at the impact of multiple sites of care on lupus attribute identification. And what we did is we broke our lupus patients up now into two populations. One that had only a single site of care and one that had multiple sites of care. When we look at the sex ratios, they're pretty much the same. When we look at the racial categories, it's similar to what we saw in that very first study I showed you, about 60, we see a skew to African-American patients in the patients with fragmented care. And we also see that public insurance skew. So kind of nice, it's repeating the results from our previous study. But what was really just fascinating to me was if we looked at those patients who were getting care at a single site that was documented for us, what we noticed, and just focus on I think the SLIT criteria here. We found that on average, those patients, we could identify 3.1 classification criteria. However, when they were getting care at multiple sites, we had the data from multiple sites, on average, we were seeing like a whole extra criteria. And of course, we did test a significance, and those are significantly different. And while we still have a lot more to investigate and dig into this data, I think this sort of is leading me to another hypothesis I want to test in the future, which is this suggests to me that for those patients who are getting multi-site care, we can in fact find more data for them. But it also suggests to me that there may be a group of physicians who are caring for these patients who don't necessarily have the data from the other site. And that absence of data could be having an impact on long-term care for this patient population. So that is a stay tuned for the next work. So the other thing this kind of tells us is more generally, more sites, probably more data to identify possible lupus. Last, just sort of quick, where are we going? Because I think every good play leaves you with some what happened and what's next. So not surprisingly, we're starting to use these classification criteria to see if we can do some clustering and identify patient populations with different outcomes. This was a quick an experiment that was done by in collaboration with Yuan Lo and by a graduate student, Yu Deng, who's now since got her PhD and gone off to work in a pharmaceutical company. But what she was able to find here by doing some clustering is that we could sort these patients out. We find a population of patients who generally have more severe presentations of severe renal disease, severe more antibody presentations that are characteristic of the disease. And we find that on this other side, this is Kaplan-Meier curve that's looking at end-stage renal disease. And our patients with the more severe phenotype, in fact, do have more end-stage renal disease. That's kind of a trivial finding, but it's sort of helping us set up a system where we can test for some of these things and look at more of the specific types of criteria. Also, I am definitely interested in the genomics piece of this. And so Northwestern we are part of the Electronic Medical Records and Genomics System network. And what's really cool is actually I've got about 200 of cases of people with lupus who also have their genomic information. Unfortunately, that's not really enough to do a good study, but when we ran this phenotype over all of eMERGE, we found about 4,200 cases. So I have a student, Noah in particular, who's now starting to explore the sort of genetic relationships, as well as think about whether we can develop polygenic risk scores using the classification criteria. And last but not least, in terms of thinking about how we might improve care overall, this is just a slide to we've been trying to understand whether we can find the data required for quality indicators for lupus. And this was published not too long ago. Actually, I should have put the reference up. But what we found is that generally speaking, most of the data is present, although some of the data like this T-score has to be pulled out of notes. And it gives us, again, sort of a foundation for thinking about how we can use data that's found in the clinical clinical record to support clinical decision support, as well as sort of new interventions for sort of improving, making sure that patients are actually getting high quality care across their experience. And just last but not least, leave you with my sort of big slide. And that is my goal is sort of an informaticist who loves the medical record system. And you can see that sort of at the very center of this picture, is to figure out how we put that clinical data, instead of sort of making it the floor of everything else, we make it the sort of foundation, the center around which we wrap many other things so that we can ultimately do disease management and modeling work. And so, we need lots of tools. I think we need good tools to standardize clinical data capture. We need strategies to identify different parts of the population, as well as sort of quality reporting tools, the clinical guidelines that we can assess. We need good libraries of tools to find patients and describe them. Ideally, we'd have a lot more patient-reported outcomes as well, because the patient perspective, representation of the patient perspective will help us, I think, come to more nuanced understanding of what's going on with people. Obviously, we need lots and lots of people with different experiences to sort of help us both develop the tools as well as help interpret what we're seeing. And we need increasing amounts of tissue samples and other kinds of omic data integration, so that once we sort of have the clinical phenotypes, we can look very, very quickly not only at genomics, but transcriptomics and epigenetics and other kinds of things that help us understand lupus. And we need to combine that with, I think, really novel informatics methods, including privacy preserving data integration and storage that allows us to sort of get data from multiple organizations, since lupus patients lupus is relatively rare and we might not have significant samples at one organization. And then we need to start developing more probably novel or machine learning based data analysis strategies that help us find signals in this data that might not be obvious from more traditional modeling work. And then I think if we do that, we have the opportunity to identify risk factors, biomarkers, and quality improvement strategies And then we can take some of those to generate hypotheses that go back into those mouse models because they're really good for testing some kinds of mechanisms. We can also figure out like what we can learn more from targeted clinical studies, data that we have in the EHR, and where it's important to get human samples. So that's pretty much it. Just like to say very much thank you to a lot of my lab and collaborators, in particular, Dr. Ramsey Goldman, who has been pretty essential for both as a clinical expert and as a mentor and as the creator of the Chicago Lupus database. I also want to highlight Abel Coe, who many of you know because of his work kind of creating the integrated data sets that I have worked with. And again, Noah, who did a lot of the work for analyzing a lot of the CAPRICORN work that I showed you today. So thank you so much. And of course, I'm happy to answer questions and also just happy to talk further outside of this if any of you would like to. And oh, I should also say, I'm also happy to share these tools if anyone's interested in using them. There's an early set on PhD. I'm also happy to share both our PowerTalks mappings and any other information we have here at Northwestern. So thank you so much for your time today.
Thanks so much for an interesting presentation. And we always, we're translational here. It's always good to see that connection between the genomic and the clinical. That was a little it was a little bit of an eye opener when you showed that slide that that diagram and in which the health data was sort of like this metadata, like bottom of the list kind of thing. I guess it all depends on your perspective, but I hope that's changing and that it would be more integrated than that. The question I have is, you mentioned that the HealthLink data repository, the Chicago one, was no longer accessible. Is that because of like funding or the local support of it, or it's like a larger national effort that isn't as active anymore, or has it just been supplanted by CAPRICORN?
Kind of all of the above. So initially it was something of a, I don't want to say it was a toy, but the primary focus of it was to sort of the first round of privacy preserving record linkage strategies that Abel was working on. And as you can see, it was really only for a certain date range was from 2006 to 2012. And one of the learnings from it was people kept talking about how are we going to update it? And essentially it required that all the data come to Northwestern to be integrated and maintained. And as you can imagine, convincing 8 large healthcare organizations to share all their data for that, it's really hard. And so I think it's sort of naturally I mean, to be honest, I suspect the data is still around. And I suspect if someone emailed Abel and said, hey, we'd like to work with it, I'm sure he would try to figure out a way to help people do that. But given that the end date is 2012, I suspect that it may or may not be so useful for certain types of assessments. But you're also right that CAPRICORN came along and did it better and solved some of the challenges that HealthLink had, which was like, again, the sort of getting all the data at one site. CAPRICORN, everybody keeps all their own data and they have an honest broker in the middle that helps with the sort of de-identification and deduplication and sort of data aggregation. And so that federated model has worked much better. So that's kind of why that, and also the data is more recent. So that's why the difference.
And the data probably is not recent because people aren't using that anymore. So also what, you were looking at association with of outcomes with patient characteristics like demographics, location, and fragmentation of care, and you also looked at some markers for diagnosis. Any work or on the horizon for looking at outcomes and interventions?
Yes, actually. So one of the studies that I didn't mention that sort of is still kind of in process is we're trying to understand that actually pregnancy is very complicated for women with lupus for a variety of reasons. They don't always have really diseases associated with much poorer pregnancy outcomes. And so one of the things we're doing right now is trying to understand how we identify pregnancy in this population and different kinds of pregnancy outcomes. And then also take a look at sort of there is a set of guidelines that are used by rheumatologists to sort of help manage pregnancy in safe ways that sort of are geared around sort of improving outcomes, medication management for the patients, also a variety of just other things that help. Again, this is where I'm frankly not clinical enough to understand all the details, but just essentially a series of things related to medication management that help the pregnancies be safer and more successful. And so we're looking at that right now, actually. And our next query for Capricorn is going to be to go back to look for that patient population. And so we're looking here at Northwestern, and we're going to sort of identify that patient population, identify elements of guideline-driven care, and then try to look at the association between guideline-driven care and pregnancy outcomes for women with lupus. So that's kind of one of the next things. And again, happy to we will probably also be looking at a few more things like cardiovascular disease and other things. But right now the pregnancy question is the big one.
Yeah. Well, it looks like you're branching out and doing the things that I guess being as a clinician I'd want to know, and also this project really highlights the importance of team science. I know that you're collaborating with clinicians and yeah. So any other questions? I don't want to hog the floor. Feel free to unmute yourself. I think we know most people here, or enter your questions or comments in the chat. Any additional questions or comments?
Yeah, I have a question. How do you define your controls in HR data? You mentioned case control, identification. How really define the controls?
Well, I mean, one of the things obviously I didn't spend a lot of time talking about, because I think with an audience of informaticists, we all live in this space, is that, obviously sometimes you don't know what's missing and what's real, right? And what's really not there. And so our controls for the middle study that I talked to you about, where we were characterizing the classification criteria, were quite literally, we looked at patients who got in care in general medicine. So they weren't just coming to Northwestern Medicine for specialty care. And we made sure that they had at least 4 visits. Again, that was empirically derived based on, we had 4 classification criteria. And we wanted to make sure that, part of the reason we weren't seeing is just that they didn't have enough visits in the system. It is really kind of hard to generate good controls, because truthfully, patients with lupus tend to be sicker, right? They're hitting the healthcare system more often. We tend to have a little bit more information about them. And so I think that's still a caveat for this. We also required for this control cohort that they did not have a diagnosis of lupus. And although that said, we actually did, as we got through the adjudication process, I think we did identify one person who did have lupus, but didn't have a diagnosis. So again, sort of how you set up controls is really, can be really challenging. That's part of why we've been trying to work in more general populations. And one of the things we're doing with pregnancy study is using our identification tools on a more broad population. And then we've been done a lot more chart adjudication. And one of the things we found is if we really want to identify lupus really well, just lupus, we probably want 4 or more diagnoses. We want one or more of those diagnoses to be by rheumatologists. And then we'd like to see a couple of particular types of labs that are really specific to lupus, such as complement labs and anti-double-stranded DNA monoclonal antibody labs. So that's sort of what we sort of refined our algorithm to for clinical data. Still all structured data, but that's the piece of it. So yeah, controls are complicated, and that's why we're trying to run this in populations that are a little less highly selected, as it were. And like I say, in the Capricorn study, our controls essentially were pretty much anybody who'd had 1 or more classification criteria, but didn't have a diagnosis of lupus by diagnosis code.
Additional questions or comments? Well, hearing none, then we're about 5 minutes before the top of the hour anyway. Thanks again for a wonderful presentation, and everybody have a great weekend and a happy Lunar New Year. And we're wishing you prosperity for the new year. All right, goodbye, everybody.
Take care. Thank you.